It's a great pleasure to be here. I'm very honored to be part of this very old and very famous academy. And I'd like to talk today about vision and how to restore vision in blind patients that I've been doing since 20 years in my career. I studied medicine and mathematics, and then I ventured out for biology. Vision starts with a video. That video is captured by our eye, and information from here flows to the center of the brain, an area called the geniculate nucleus, and then arriving to the visual cortex, the primary visual cortex. From here it radiates through our brain, our cortex, and in humans, around 50% of the cortex, we can measure visual activity. So humans are very highly visual animals. So this is the cortex. And in my career, I've been studying how the retina as a computer pre-processes images, how then the LGN makes sense of it, and how then finally the cortex is preparing it for forming a person. But today I don't like to talk about the basic investigation, how this computer works, but rather about applying this in diseases which result in complete blindness. And I'll show you our attempts in the last 20 years to restore some visual activity in diseases, in a variety of diseases that leads to blindness. So one thing to know about blindness, almost all blinding conditions originate in the eye, and those that are unsolved are coming from the retina. And the retina is not a simple light detector, it's a biological computer. It has one input, that face of Roger Federer, you see it, and it generates around 30 different movies, features, pulls out features from the visual scene that is broadcasted then via the optic nerve to higher brain centers. And if you look into the hardware, and there's a drawing by Ramon y Cajal, a famous anatomist many years ago, he already noted that it's a very special computer. In the computer you use, it's in your pocket, you have only transistors. But the retina and the brain in general is built differently. You have many, many different kind of processing elements. Indeed, you can count around 140 in your retina, and these are connected together. So everything starts with photoreceptors. This upper layer are photoreceptors, and then they talk to so-called bipolar cells, the ganglion cells that form the optic nerve, and in between, there's a massive amount of computation giving rise to this 30-feature extraction I showed you before. Now, the dilemma for diseases, for a number of diseases, is the photoreceptors lose their function or they die, such as macular degeneration, age-related macular degeneration, and you still have a computer, but you have no input. And what we tried to do over the last 20 years is that somehow bring back photosensitivity to these retinas and use the internal power, computational power, then let patients to perform visual tasks. So the diseases we are working on are partially inherited diseases caused by 280 different gene mutations called IRDs. These are usually younger people. Or on geographic atrophy, this is the end stage of age-related macular degeneration, which results in central blindness, and the first result in complete blindness. So the therapy, the story of the therapy I started to talk about started in algae. And why algae? Because these are very simple organisms that can also detect light with a single gene that codes for a single protein. You need around 20 proteins to code for your visual sensitivity, and we have no technology to bring back 20 genes, but we do have technology to bring back a single gene to somewhere in the retina. So we decided that we take these genes from algae that make light sensitivity and try to bring it back to human patients. And so this is the idea that you start with a single gene encoding for a light-sensitive channel. This is a channel which opens, and the voltage then pulls in current, changing the photoreceptor's memory potential, and try to target it into key elements of this 140-element circuitry to bring back some interpretable vision. Indeed, there are 4 different potential ways of bringing back vision, targeting different cell types. So one, you can target, if there's still photoreceptor elements, you can target them even if they're blind to make them light sensitive. If they are missing, you can go to the second layer, the third layer, or the fourth layer in the retina. So the technology we are using is gene therapy, a form of gene therapy in which you use a virus called adeno-associated viral vectors. It has been virus one time. Now it's a completely synthetic device. There's nothing, cannot replicate. It's not like COVID. This is just a little ball, what we call a capsid. It's 20 nanometer diameter. And inside, there's a single-stranded DNA. And the DNA has two elements. One is the channel that we are encoding, the light-sensitive channel. And a second element, which is a postal address. It has an amazing capability that the viral vector will flood the entire retina after injection. But this little postal address, we call it promoter, will only allow the gene expression in the cell type that we decide to do it. It's a technology that took more than 15 years to build up to get the right postal addresses. But today, with exquisite precision, I can inject a virus or a viral vector into the eye of a patient and can tell which cell type to express whatever protein one, in this case, is a sensor. So briefly, I don't want to go too much into detail, but the way we plan and engineer these postal codes is by taking human retinas post-mortem, taking them to single cells, hundreds of thousands of single cells, and then measuring gene expression. And from that, using a large number of computer codes, we can pull out essential elements for this postal address, and then coming about 1,000 different potential viral vectors. To test these viral vectors, which one would work, use a technology that we can keep now human retinas alive after the death of patients for at least 20 weeks. So these are retinas post-mortem, and we can test our gene therapy vectors, several hundreds of them, or up to 1,000 in them. So this is how we build gene therapy in actually a human tissue. So we find vectors, and here's just examples. This green dot shows you which cell types we are targeting, and one by one we can pull out viruses that, or gene therapy vectors that target the right cell type out of these 140 that we want. So the first therapy I'd like to show you, historically we started with this one. This was the simplest. We started around 15 years ago together with José Sahel, in which we target ganglion cells. So ganglion cells is the last layer in the retina. So if you target this cell type, all the computation in the retina is lost, except you create a light-sensitive layer. The computation is lost because it's the last layer that you target with the light sensitivity. So basically, information directly mapped to the brain. So for this, we do a simple injection into the eye with this viral vector. And then we build for patients a special goggle, which is a video camera watching the outside world, and a little projector in the back that shows the light in the right wavelength we need. And also, we do a pre-computation. Since the retina lost its capability to compute, we pre-compute the image that will be shown to the ganglion cells. So let me show you two examples of patients. The first patient was 58 years old. It's a French patient who lost vision 10 years before. And we do this injection of the therapy. We train them to use the goggle. And we have two conditions. One condition, and the goggle is not on. And under this condition, it's like normal, it's like the blinding condition. They cannot perceive the world. When we put on the goggle, we use a special stimulation that they can sense with these algae light sensors. And then we evaluate what sort of behavior they are capable to. This patient has a so-called bare light sensitivity. That means that that patient can recognize where is the window the light is coming from, but unable to see objects. And the task you will see is to perceive, to locate, and to touch the objects. So here the first test is that when it's called natural monocular, so the goggle is not on, and this is the background condition, where the task is actually to find that notebook. I don't know if we have sound. We will see in a second.
Enfin, je perçois. Au bout d'un certain temps, je ressens qu'il y a une différence d'intensité.
So the patient finds that there is an intensity difference, but cannot detect the object. And now we put on the goggle that should now enable this restored vision. And you can see what the camera sees at the bottom on this computer. So the patient is scanning the field with the goggle. You can see this horizontal scan until a point where the object is now on the camera.
Oui, c'est là.
Ok.
That's a perception.
Et alors, c'est où? C'est...
Ah, pardon, c'est sur la droite.
Ok.
This is the localization. And this is and watch the motor action with incredible precision, the motor action 10 years after the loss of vision. So what we see, this therapy only makes a small light sensitive area in the retina. It's a tiny, tiny area. So it has to scan the field, but so it takes a long time to find the object. But once it finds it, the internal motor program is immediately activated and with exquisite precision can point. Now I show you how really precise it is in the next patient. She's 70 years old, no light perception at the treatment, and she has the following task, is to count pens on a table. Again, you can see the goggle, you can see the objects on the camera, and she's counting 1, 2, 3, the pens. So, 2, 3.
Et le bord de la table du fond est où?
I have to find the edge of the table, and then again the task is to point at the pencil or the pen and look at the precision. So what we see again is it takes a long time to detect the object. It's partly a mental calculation because they don't have a really good vision. It's just bumping to objects. But then using this ability to start the motor program, which is always very precise. So what we have learned from 5 patients over the last few years is that they have that the treatment has very good safety profile. There's a good object recognition, but no face recognition and no reading. And so the question, why is there no reading capability? And to explain that, I like to point your attention that human vision is very different from most animals' vision. We have a special area in the center of the retina, which is called the fovea or macula, which is for high resolution. It's like a very high resolution pixel density area there with a very specialized circuitry that only exists in monkeys and humans, and no other mammal have it. And in order to read or face recognize, we always use that. Indeed, patients who lose at very center 1% of the retina, they think they are blind, and patients who lose 95% of the rest, they are perfectly fine. So we humans really rely on this central area. And what happens is that in this area, there's a very special geometry of ganglion cells. They are on a torus. So what happens if you have a torus light sensor? You show an image of a face, and with this torus, there's nothing in the center, no ganglions at the center. Therefore, you will not see the nose of a person, and then the rest become distorted. So once we realized that, we started to build a new therapy. We call it Optogenetics 2.0. This is shown the lower one. If we target now not the ganglion cells, but the cones, they are on a mosaic, and there's a potential to restore really high-resolution vision. This is our next goal now in clinical trials. So very briefly, I'd like to show you how now we develop in a human retina this new therapy to target a particular cell type called the cone cells of the retina. So it takes around 4 years to get this postal address. But once we have a good postal address, we take these human retinas, we drop them these gene therapy vectors, and all cones, we can target the gene and no other cell type. But then we still have to evaluate before therapy, is there any function of the retina? So what we use a technology that we can take these human retinas, and the very first day after the death of patients, we can indeed measure activity from the retina. We can measure the same activity as normally the ganglion cells via the optic nerve sent to the brain. We can measure with a little chip that has 30,000 electrodes. We can put on the retina and stimulate with light. And doing that, here you can see that actually an hour after the death of the patients, we can see the entire retina as we flicker light responding to it. And then we can measure these information channels. There are 5 main information channels in the human retina. We can measure that, how they respond to a pulse of light. So here it shows you to turn on the light, you turn off the light, and each of these 5 different information channels respond differently. Now what we can do is that we take the same retina and culture for 5 weeks. And the first days, this human retina loses light sensitivity for unknown reason. we apply the therapy, bring back line sensitivity, and 5 weeks post-mortem now, we compare what sort of artificial vision we can bring back. And what you can see here, if you compare the left and the right column, is almost identical human retinal output we can create now with the cone-based light sensors. So one main question for us was, are there patients with remaining cones? And if there are, how many of them? So what we did in our institute, we went around 18 countries around the world, collected data from blind patients using so-called optical coherence tomography, so we can measure the presence of cones in around half an hour in anybody who walks into an ophthalmology clinic. And we found that around 62% of the retinitis pigmentosa blind patients, the same with macular degeneration, retains these cones. So these will be eligible for this new therapy we are building that we hope to restore some ability to read and recognize faces. This is my last slide, which I like to sort of circle back from medicine to basic science, is that what we have done in the last 20 years is build therapies based on the knowledge we learned, how the retina works and higher brain centers, and use biotechnology to build light sensor in the retina. But now with this therapy, we learn something new that really helps basic science. Namely, we can make a human retina in a dish way after the death of patients light sensitive. So we have a completely functional human brain part in a dish for several weeks. The very first time in human history, we can start to study how human systems compute information. And this is what we are doing now currently in my institute, in my laboratory, to try to exquisite precision to understand how human retinas perceive and compute visual inputs, and how it relates to the percept we form as you watch me talking. With this, I'd like to thank many people in my lab, in my institute, and my collaborators, especially José Sahel, Hendrik Scholl, and Bence George, with whom I worked together for a long time. And thank you very much for your attention.
Thank you very much for this impressive lecture, for the very, very nice result. But I have a few questions. Let me start with one. At the end of your talk, you were mentioning that you were going to study in details how retina is working. I mean, I think, but I would like to know if this means that you are doing some kind of multi-electrode measurement activity on neurons, and I wonder how many neurons together you can measure, and how all these things are done, and how all the huge amount of data, I believe, that come out from this measurement are analyzed.
Yes. Because
this is a central problem, I would say, and also to understanding, because it's very important to understanding how natural neural network, natural neural system behaves compared to the artificial one, and the one that I believe that is quite different the way that they work.
Yeah, they work very differently. So first about recording, that we used to do in the last 20, 25 years, we have been recording from mouse retinas and animal retinas. And the key, which we understand quite well, so the key to understand how the human compares to the other animals, we use many, many technologies to record. So multi-electrode array, we can record, on a good day, we can record maybe 10,000 cells. We now have voltage imaging, so we can put genetically encoded dyes into the ganglion cells, where we can, again, image activity. There are many, many methods. We have electrode arrays, so there are 150 people in my institute who are working together, and it's a huge amount of data, so much that in the institute we cannot store it anymore on our servers. So we use Google so basically we cannot compute anymore locally. We can only compute on the cloud. because we have to invert matrices which are 20,000 times 20,000, and that's just impossible with any normal computer. So computing and more mathematicians we hire because it is becoming a data science exercise.
Yeah. Domande? Sì. Stefano. Vabbè,
Yes.
qual era la prima, scusate?
I think there was somebody.
Stefano Scaffino, mi pare. Ah, devono prendere un po' di altro.
What about artificial retina? I know there are groups using an alternative approach compared to yours. Can you comment on that?
Yeah, so that has been, before optogenetics, there was a long time people built artificial retinas. The problem is resolution. The problem is that the resolution of individual cells of a cone photoreceptor is 4 micron. And when you talk about these electrodes, there are hundreds of cells behind it. That's one problem. A second one, it's a very complex surgery, why this is just an injection. So most patients, they ask to be removed after the surgery they have the retinas because they don't see enough benefit for that. So I think what brings the future technology, currently these arrays are very low resolution and very expensive surgeries as compared to gene therapies.
Thanks very much. Very impressive approach, especially on the recording of, expianto retina. Still, to be viable, this approach needs to be free from the goggles.
Yeah.
You need to introduce eye movement. So what you think, can you excite the virus anyway?
Yeah,
the second question is about really, could be a central gate, or at least also we have a demonstration of a central gate that after many years of blindness is forbidden the brain to re-elaborate incoming route. So how much time do really we have from when applying the technique? Thanks very much.
Very good questions. Very good questions. So the first one is that the first technology I showed required the goggle. The new one with the cones, they don't, because we were able to improve the sensitivity that in a bright room, these patients were able to see. Now, the dynamic range of all light sensors is around 3 log units. So we are able to tune so our first aim is for blind patients give the ability at home environment to function. We don't believe that in the next few years they will be able to walk out and be alone, autonomous, but they will be able to, that the goal is to be able to function home, and that we can control lighting. So that's about the first part, that as we move. The second, that even with this new therapy, there are new technologies with goggles, and we have now scanners that can find the position of the eye. And then we have a new technology to show a much larger image and know where the eye is positioned, and we can only light up those pixels. So that's, but I would say that the more promising one is that when you don't have to wear the goggles, and this is coming at the next generation. Now, regarding the gait, what we see is that it depends on people. So some of the patients we have are for 3 years able to recognize objects. So it doesn't seem to, at least we don't see that gait that they would going down. Now, it is true that some of them requires extensive training at the beginning. So very, very much like is that in the 10 years, this cortex has been started to do all sorts of tasks, and it needs training to regain, to actually concentrate on vision. So far from 5 patients that has any ability, what we see that they improve over time, and then there's a certain amount of ability they have, and that's it. So we believe at this point is that ability, the limit of the ability is because of the strange arrangement of the ganglion cells in the fovea. But with the cone-based optogenetics, we will learn that is there any really hard limit that is imposed by the brain and that we cannot yet predict. And there might be.